right?
Yes.
Okay. Now if you notice what is happening in A, they have given a nucleus and there is something like this. B, you have the set of chromosomes, right? This is A.
Mm-hmm.
What is wrong? What is wrong? Okay, one moment, one moment. This is A. Okay, don't. Oh God. A. B. B shows you a number of chromosomes. See what is happening is it's actually dividing like this. Studied the process of mitosis earlier.
Mm-hmm.
Have you done it in school? Okay, so here we have C, we have D, and we have E, right? Okay. Question is
Mm-hmm.
A is preparing to divide. Okay, they have given you a hint that they are preparing to divide. What is happening to the cell? And we have to tick two boxes, right? Okay, we'll see one by one. The nucleus is dividing. Can you see the nucleus being divided here?
No.
No. Okay. The cytoplasm is dividing. Can you see the cytoplasm dividing?
No.
No. Okay. Cytoplasm is not dividing, nucleus is not dividing. Then what is happening?
Um.
Next option is
Crop.
the number of subcellular structures is increasing, right? See, they are increasing. They have given you a combination of something which is like kind of multiplying, okay? So we'll see one option of that, okay? We'll take out that option that the number of subcellular structures is increasing. Then we have is the chromosomes are doubling. Yes, here the nucleus inside them, what is happening? The chromosomes are doubling. Only after the chromosomes are doubling, you will get this figure B. Okay. The last option, the chromosomes are splitting, that is happening only in the figure C. Okay. Any doubt?
What was the other reason why for
Sorry, what is that?
what was the other reason?
What are the reason for?
Um, it says tick two boxes.
This one, the E part?
Which one?
What are you asking about?
1.3. It says tick two boxes. Which was the other one?
Which one? Which box are you asking me? That only I'm asking you. Which one? Can you point it out that for
Oh
me?
no, what about 2.3?
Option 3. Okay.
Uh-huh.
The first one is was the number of subcellular structures is increasing, right? That and the is the chromosomes are doubling. That's what I said. Only when the chromosomes are doubling in this part, we will be getting this structure. That is a B1.
Okay. Uh,
Clear
Fish.
this part?
Yeah.
Or do you want me to explain how mitosis works? Tell me that.
Or how much is
You want me to
um
explain mitosis to you?
pardon?
Do you want me to explain mitosis to you?
Oh no
It's fine?
yeah.
Okay. Question number 1.4. Describe what is happening to cell D. Okay. What have you
The,
done is the cell has made a copy version of it. Okay. Here the nucleus is dividing, right? And okay, you can just say the nucleus and the cytoplasm both are dividing. Okay.
okay.
Nucleus and cytoplasm dividing. Okay. The answer what you have written, the cell has made a copy version of it, will be applied to figure E, not to figure D. Okay, because only in figure
Oh
E we can say that both of them have separated, right? But here they are still on the verge of getting separated. Clear?
yeah.
1.5. How do cells produce? Okay, how do the two cells produce at say stage E compared to parent cell A? Okay, then they have given you three different options. They are genetically different, genetically similar, genetically identical. Yes, the answer is right that they are genetically identical.
Mm-hmm.
So you're clear with chromosomes and mitosis?
Yeah.
Okay. Can next one? You're done with this, Jad? Can we
Yeah, okay.
next one?
Yeah.
Okay. Now this is regarding binary fission. Okay. First of all, what do you understand by the term binary fission?
Um, sure.
Not sure of what is binary fission?
Mm-hmm.
Simple terms I will tell you what is binary fission. Okay. If I am having a cell, okay, it
Okay.
has nucleus, it has all the things inside the cytoplasm. Same kind of mitosis is happening here. Okay, what happens here? Separation takes place. There is a nucleus formed in one end with these cell membrane cell organelles. So this will be like that. And then you have two different cells. Understand? It is just a division of a single entity into two or more parts. Okay, and here also again they are going to resemble the original one. Fine. So this is a binary.
Yeah.
Okay, going back to the question. Question number 1 says scientists performed an experiment to observe the growth of bacteria in a nutrient broth at 25 degrees Celsius. Okay, so they have a nutrient broth at 25 degrees Celsius. They have given you a table in which time and absorbance is mentioned. Yes. Use
Yes.
table 1 to finish plotting figure 1 and draw a line of best fit. They have marked, is it you which have you marked or is it marked earlier?
Huh?
Crosses.
Well, marked it.
That is your marking.
Which,
No, what I can
sorry,
see,
which question?
see, binary fission, that chapter.
No, I have marks here.
It's not your marking.
Which full?
No,
Is it
because
1.1?
the graph, they have marked some
Oh,
crosses
yeah,
over
yeah,
it.
yeah.
Is it you or is it marked?
Yeah,
No, right?
I'm up.
So they have given marked 2-1. Let's draw it again. Let's see. So what have they marked is they have marked a point 0, right? According to the table, 0 is at 0. Then 0 and 0.5. So it will come somewhere here. Okay, they have marked this one. Then points have been marked.
Yeah.
Right, if I'm not mistaken. Okay. Okay. And we need to mark the rest, right? Use the data in table 1 to fill
Yes.
the figure 1 and draw a line of best fit. So can you tell me which one will be? They have given the second one, fourth one, for time 4, that is for minutes, for hours. For 4 hours, what is the observed value absorbance? For 14 hours, 16 hours and 2 hours, they have already marked. What is the value for 4 hours? What value can you see?
0.10.
1 point. So it is
0.10.
okay. Again, this will be somewhere here. Plot will be here. At 6 hours,
6 hours would be 0.2.
okay, exactly at 2, right?
Yes.
Okay. At 8 hours,
8 is 0.4.
0.4.
Uh-huh. 40.
Okay, so it will be this way. Okay, 10 hours will be
10 would be 0.66.
0.6.
6.
6, right?
Uh-huh.
0.06 will be somewhere like this. And for 12 hours it will be?
Or would be 0.78.
Okay, so 0.8 again. So 12 will be here again. Okay, So my points are fixed now, right?
Uh-huh.
Understood how my points are going? Now our job is only to join them. So we have to join all the center ones like this. Okay, so it will be somewhat like this. It will be a good curve. Okay, if you're drawing it in a book, it should be a good curve over there. Here, first one,
Yeah.
have you plotted it down?
Yes.
We'll move on to the next question.
Yeah.
The absorbance increases because the bacteria are dividing. What type of is occurring? Okay, we are talking about binary fission, right? So my
Uh-huh.
answer will also be binary fission. The cell of how it is dividing, the cell type of cell division is nothing but binary fission.
Uh-huh.
Clear?
Yeah.
Next question. Suggest one factor that might be limiting the population growth after 14 hours. Okay, after 14 hours it has been constant, like I mean, if I can say after 12 hours itself, it is remaining constant for 14 hours. And why is that so?
Why is that also?
Why is it being constant? If you observe after 12 hours, my absorbance rate, this is absorbance, this is remaining constant, right? It is not increasing nor decreasing. Why is it like that?
Um, not sure.
Okay, what are we talking about here?
Huh?
What are we talking about here? We are talking about binary fission, right? And we are talking about the absorbance rate. So what we can say is that why this has been constant here, because the amount of nutrients or amount of oxygen suggests one limiting factor. This might be the limiting factor. That means amount of nutrients or the amount of oxygen is being constant. And therefore my absorbance is also constant. Okay.
Okay.
That is 1.3. This is answer 1.3. Okay, moving to the second question. A type of bacteria was found to have a mean division time of 45 minutes at 20 degrees Celsius. If one of these bacteria was given the same conditions, how many bacteria would be there in the population after 9 hours? Give your answer in standard form. Okay. Understand the question first. Okay, what have they given? They have type of bacteria which has a mean division time. So we have mean division time is 45 minutes. Okay, and that is at Celsius. If one of these bacteria was given the same conditions, how many bacteria would be there in the population after 9 hours? Okay. Now we have to find the population after 9 hours. Okay. So how are we going to do this? First of all, we need to convert this 9 hours to minutes. So how can we do that? 9 times 60, right? 6 times 9 is 54, 4, 5, 40 minutes. Okay. Now what they have
Okay.
mean division time, that is 45 minutes. So we will see how many divisions are possible in 540 minutes, right? So simple addition, 540 divided by 45, which gives us to the value 5 nines are, 5 ones are, 5 0 eights are, 9 ones are, 9 twelves are, right? So 12 divisions. What are we getting? We are getting 12 divisions in 540 minutes. That is in 9 hours. I am getting 12. But what are we supposed to find out? We are supposed to find out how many bacteria would be there in the population. We have to calculate the number of bacteria, right? So we are going to put this 12 in, okay, bacteria, second bacteria. So 2 times 12. Okay, either you can stop over here saying there are 12 divisions and 9 hours happening. Okay, so according to time, what is 12 to the power 12? 2 to the power 12.
Twelve to two to the power 12.
2 to the power 12. So you have to multiply 2 12 times. 3, 4, 5, 6, 7, 8, 10, 11, 12. 8. 8 was a 16. So 16. 16. 16. 16s are
16 times 16 is
16. 16s are 256 times 16.
4,096.
4096. So this much amount of bacteria is being produced now. 4096 bacteria.
That's huge.
Okay. You understood how did we get this value?
Yes.
Any doubt over here?
Huh?
So shall we move on to the next question?
Yeah.
Now next is regarding culturing microorganisms. Okay. Page number 8. Okay, this is entirely a practical method. Okay, fine, we'll do it. A student was investigating the effect of different concentrations of an aseptic on the growth of Bacillus subtilis bacteria. So they are going to conduct an experiment on a bacteria named as Bacillus subtilis. Okay. The students prepared uncontaminated cultures of the bacteria on agar plates Why was it important that the cultures were not contaminated? Why do you think so the cultures were not contaminated?
Um,
Tell me what will happen if the cultures were contaminated?
it will, I think, will. So,
Tell me then what will happen if it gets contaminated?
not sure.
Not sure.
I'm not sure.
Okay, wait. Now this is a plate, okay? And they have made a smear of bacteria. Fine. This is the way they make a smear of bacteria. Now they are supposed to grow Bacillus subtilis, right? If it is getting contaminated, what can we observe that? First of all, it will affect the result of the experiment, right? We'll also see that we have some growth of pathogens. What do you understand by the word pathogens?
Um, I think it repeats itself.
Sorry. Pathogens, I asked you. What pathogens?
Um, I'm not sure.
Okay. Pathogens are microorganisms which cause diseases. Okay.
It's,
Microorganisms which cause diseases. So there might be some pathogens which might enter into this. What are we preparing? The culture, okay? The bacteria culture. So this is answer 1.1. 1.2. Suggest three things that the student may have done while preparing the plates to prevent contamination of the cultures. So how could the student prepare a culture which is not contaminated? What are the possibility methods the student might have used?
I'm hoping to.
Okay, I want to prepare this plate of Bacillus subtilis, right? And I want to make
Hello?
sure, okay, just imagine that I have to prepare this plate of Bacillus subtilis. Okay, fine.
Uh-huh.
Now I have to prepare a very pure form without it getting contaminated. So what measures I can take to make it look sterile?
Maybe not putting thing over it. That I
Yes,
think
and I'm waiting for your answer.
I think not putting the name over it.
Sorry, what was it?
I said not putting over it.
Okay, before that, we have to sterilize whatever we are using. Okay, first method, sterilizing. We have to sterilize starting with the petri dish to the, the loop. Everything we are using should be sterilized.
Uh-huh.
Then what can we use is yeah, that is all one, like sterilizing, then putting the culture medium with a used sterile.
Okay.
Okay, sterilize will includes all your equipments which you are going to do with the plates. Okay, so next question. The student prepared 4 different concentrations of the antiseptic by diluting the original solution. She labeled them A to D as shown in table 1. The student soaked a small filter paper disc in solution A and placed it carefully on one of the prepared agar plates. She repeated this with a solution of A, B, C. And the plates were incubated at 25 degrees Celsius for 2 days. Okay. Why is it important that the birth rate weren't incubated at 25 degrees Celsius?
Um, why is it important?
Big question, yes.
Um, hmm. So I think where it's like dry out or here.
Okay, you understood this part?
What part?
2.
Why is it different?
Okay, which one? 1.3, right?
Yeah.
Important that the plates weren't incubated at 25 degrees Celsius. Why? The only answer we can give is because we can reduce the chance of them getting harmful pathogens, right? We have to remove pathogens, so we can include this answer to that. 1.3 will say to reduce the chances the growing pathogens. Okay, we'll move on to the next question.
Yeah.
So they have given you a table and they have asked you to calculate the value of x. Okay, where is x? It is there in the column B. Can you see it? Mean value. I think that you can solve it. The mean value, to find out the mean value.
Of B.
Yes. Of
The mean
solution
value.
B.
Uh, um, um,
How do you find the mean value?
um, uh, is it? Wait. Mm, 13.
13. How did you get the answer as 13?
I added all the numbers up, then divided 3. Um, how I got the answer is adding all the numbers up on the divided by 3.
Okay, that is a perfect one. You have to just add up and divide it by the 2 one. Okay. Last question after 2 days. Okay, the next one. I think that is also fine. No, we have done x, right?
Yeah, for 1.4, right?
Yes, 1.4. The results are summarized in table 3. Calculate the value of y to complete table 3.
Uh-huh. 1.5. So I can take it in my calculator.
Yes, you can do it on your calculator.
Okay, thanks.
From the answer.
Answer is 7.85 centimeters.
1.87.
No, 7.85.
How did you get that?
I squared the 5
Okay.
and times it by pi, which is 78.5. Then turned it into centimeters, then I got 7.85.
Okay, fine.
Yeah.
Yeah. How did you find out? With the way, how did you find out the answer?
Um, well, times, well, I squared 5, which is 75. Then 10 times the pi, which is 78.5. Then turned it into centimeters, which is 7.85. Yeah. Hello?
Yes. That is your.
Yeah,
Okay, let me see how.
yeah, I,
Yes, let's see how is it done. Okay. What have they given
oh, okay.
us? Okay, what are we doing? We are doing the next set, right? Okay.
Uh-huh.
Formula pi r square. They have given us pi r square, that is 3.14. Okay, fine. So we'll move on to the next one. Use a graph to estimate the area of the populations for Savannah and with the
Yeah, 1.6, you, you could put the beam radius at the x-axis, y as the area of inhibition zone as the y value. Hello?
yes. So what can we write? Only that much is enough.
On the x-axis, x-axis,
X-axis. You have to plot a graph, right?
yes.
Okay. So
Um,
yeah, just let's see how our points will be there. Your inhibition, right?
mhm.
And here will be the concentration. So, table 3, 3.8, 2.0. Okay, have we found out the value of uh, x?
What, for y,
Yes, y.
7.8, 7.8.
7.8. How is it possible? 7.8. C. Calculate the value of y to complete the table. Use the formula pi r square they have given. Give your answer in centimeter. Okay. Now, here, if you see, earlier they have given y is mean radius is 5 mm, right? Mean radius is 5 mm. Now this 5 mm, if you need to convert it into a centimeter, I have to put at 0.5. And with this exact help of mean radius, we are going to find out y. So 3.14 into the radius. I have got the radius, that is 0.5 the whole square. Now tell me what is your answer?
7.85.
7.85.
Yeah, or 7.9 if you got,
Okay, one moment. 0.5 into 0.5 will give me 0.25, okay? 3.14
oh,
into
really close.
0.25. Now calculate. It's giving me 0.7. My value is coming as 0.7. So that will
Oh, wait,
be
it will double check. Because I converted it in from millimeters to centimeters. After
you have not
from
converted from millimeter to centimeter, right? Okay.
sorry. Yeah, 0.7.
0.7.
Yeah.
Okay. So we have found the value of the area of inhibition of y, that is 0.7, right?
Uh-huh.
I have value. If this is my concentration with, let's start with 10, 20, 40, 60, 70, 80. I think it won't come. Let's check. 1, 2, 3, and 4. Okay. Of the solutions. So first one at 100 will be somewhere here. My inhibition will be 3.8. So it will be coming somewhere here. Okay, shall I draw it on the next slide? I'll draw it.
Yeah.
I'll draw it perfectly. Next slide. Okay. So we were doing 10, 20, 30, 40, 50, 70, 80, 90, and 100, right? And here of 1, 2, 3, 4, 5. This will be my inhibition zone, and this will be my concentration, right?
Uh-huh.
For 100% it was 3.8. If I had to draw 3.8, it will be something like this.
Oh.
Right?
Yeah.
Okay. Then for 2 it was 50%. For y, because of y 0.7, right?
Mhm.
Maybe somewhere here. That gives me 25. For 0.5 it is 12.5%. Right? Now let's draw the curve. So it will be what like this. If I need to draw a straight line, it should fall this way. No, I'm not getting a curve like this. Let's draw a curve. Okay?
Oh.
We'll be plotting a graph. You understood how we have got that?
Yeah.
Next one, use a graph to estimate the area of inhibition zone produced by a solution of this antiseptic with a concentration of 80%. So if T, let's draw the approximate value we might get, okay? This way, approximately it should be around 3 to 3.5, 3.4, say it like that, okay?
Okay.
3 to 3.4. So that will be my area of inhibition of 80%. Describe the relationship between the concentration of antiseptic and its effectiveness at preventing bacterial growth. Okay. Now what can you write about bacterial growth?
When it goes up, wait. Let's wait. As it goes up, the more bacteria is made.
More bacteria are made. Yes, complete. We should have a perfect answer, right?
Yeah.
Questions you can give me regarding this.
Okay.
Nothing?
Yeah.
What can we see here is that bacteria, they develop a resistance to antiseptics, okay? Bacteria develop resistance to antiseptics. Also, we can say that these antiseptics are very effective when we are using in a proper concentration. So if we are having a proper concentration of antiseptics, we can actually kill the bacteria. Okay, we can actually kill the bacteria. But if we use another concentration of very high or very low, it can only damage the tissue part. You are understanding what did I write here?
Yes.
Okay, shall we end up here? I have another class.
Yeah. Okay. Bye.
You understood what we have done till now?
Yeah.
Okay, then fine. Thank you. Bye.
Bye.